Good evening, one and all. I am Dr. Kesha Jani from IIP Digital Media team will be hosting your today's webinar on rheumatoid arthritis and in-depth psychoneuroimmunology analysis with our resource person Amaloa Casanova ma'am. The moderator for today is Dr. Zubia Wakar ma'am. This is our second webinar in the international webinar series being organized by the Indian Association of Physiotherapists. IIP is the oldest and largest association representing physiotherapists in India since 1955. The association's digital media team consists of a group of physios who handle Zoom platform, webinar live streaming, and IIP profiles on social media platforms like Facebook, Instagram, Twitter, LinkedIn, and YouTube. Follow our IP profiles on these social media to receive notifications for our events such as webinars, workshops, CMEs, conferences, health camps, blood donations drive, and to know the recent updates on physiotherapy. Subscribe to IP YouTube channel for notifications of our live webinars. In our playlist section of this YouTube channel, webinars are segregated into various folders on topics like orthopedics, neurology, sports and fitness, pediatrics, cardiopulmonary, women's health, research, cancer rehab, etc. The social media links will be shared in the chat shortly. I'll hand over to Dr. Zubia ma'am. Thank you so much.
A very good evening to everyone present in this webinar today. I, Professor Zubia Wakar, on behalf of IAP, would like to welcome all of you to the second lecture in the international online webinar series being delivered by Professor Amaloa Casanova Mendez on the topic rheumatoid arthritis and in-depth psychoneuroimmunology analysis. This lecture will be exploring the multifaceted factors underlying rheumatoid arthritis. In India, the topic is fairly new and a lot of us, in fact, are not very familiar with it. And I really hope, and I'm looking forward to a lot of new information. Rather, I think most of the information would be new for us. So I think Dr. Casanova in a good place. You don't need to worry about too many higher level questions which are going to come to you. And as far as the questions are concerned, we will prefer that you type the questions down in the chat box, and I will be taking those questions at the end of it. Just to introduce the speaker briefly, I know there's loads and loads of papers and articles available about her, but just to give you a very, very brief outline about her, our honored speaker, she graduated in physiotherapy and has 23 years of clinical experience, and she specializes in the rehabilitation of chronic pathologies. She's a PhD in clinical psychoneuroimmunology from the Faculty of Medical Sciences, University of Granada, Spain. She also has an official master's degree in social communication of scientific research, along with a master's degree in clinical psychoneuroimmunology. She's an organizer and mentor of research health programs such as intermittent living. I invite you to deliver your lecture, ma'am. Over to you.
Thank you. It's my pleasure. It's a big pleasure for me to go all the way along and to go to be in India. It's a dream for you, so I'm so happy to be here. I hope I can answer all the questions you have at the end of it. So I'm going to start. What we are going to do is to, I hope this provides an overview. Of course, it's a very complex immune pathology. So of course, we are going to mention as much things as possible in an hour to give you strategies and to give you tools to treat or to give company or to improve people with rheumatoid arthritis based on psychoneuroimmunology. So, well, first is important, we are going to focus on epigenetic environmental triggers or lifestyle changes that really are or could do a profound difference in the patients with rheumatoid arthritis. Then we are going to intense our understanding about the pathogenesis and discover innovative, I hope is a very innovative therapeutic tools and management and actually reasoning to do the diagnostic and we used to call it in psychoneuroimmunology, the film and the movie. So now we are speaking specifically about rheumatoid arthritis, but of course you have to zoom or to add many different risk factors, characteristics, environmental triggers, and specific, for example, sex or gender differences to then come up with a symptom or an inflammatory process. So, of course, what we are going to do is to speak about many of them, which are very useful for us. So what we are going to find in these patients are persistent joint inflammation. And if it's too aggressive, what is going to happen is that the immune system will be so aggressive that we can lose tissue and tendons and cartilage until the whole, till the whole joint is destroyed or sacrificed. So, of course, we have different genetic and environmental and lifestyle factors, and they are crucial. So, first, I want to make very aware that, of course, we have different polymorphisms or genetic characteristics that that are related, or we have studied in science in general, in different science areas that are related, and it is around 100 different single nucleotide polymorphisms or changes in our gene characteristics that are associated with rheumatoid arthritis. The thing is that even though, of course, that is kind of a susceptibility, there are a lot of people that carries these polymorphisms and they do not have a problem. They don't have any problem in their joints. So it's important that we take into consideration that if you are genetically, or if someone is genetically susceptible, the impact of the environmental factors is going to be very increased. The second thing that I really think is important, first thing, is not obliged and is not completely determinant. If you have a polymorphism, you are not, it's not necessary that you develop the pathology. This is a very first one. Second one, it depends on how much different susceptibilizing factors you have. If you have many aggressive genes, of course, the control that we have to have and the tools and all the different aspects that we have to treat, even medical things and medical assistance will be needed because the aggressiveness of the immune system is going to be a lot higher. But of course, then we can have a smaller or a lighter susceptibility, and you don't have any need to develop the pathology. Then, in a second hand, it's important that we understand what these polymorphisms are for. Because, of course, a few of them are just characteristics, are just characteristics of our immune system. What I mean with this, look, it's very interesting that what we call a polymorphism is a change in an immune protein that are human leukocyte antigens. So, for example, when we have a difference or a change in the sequence of the nucleotides, or exactly is not the same structure, so one of them changed, you can have a gain of function or a loss of function, but normally what we have is a characteristic of the immune system to be a lot more efficient for some bacteria or virus or protozoes or some other microbes. So 95% of the polymorphisms in the human species, belongs to the immune system. What does that mean? That means that we have been in an evolutionary process taking good care of all the environmental pathogens. We have died. One of the first causes of death in the human being have been the infections, because now we have hospitals and antibiotics. But in the past, in an evolutionary perspective, we have, walked around the whole world, and we have migrated very distant places. So of course, we found different microbes, and we have to do some polymorphisms or mutations in order to be adapted to that kind of microbes. So we have a lot of evolutionary. In evolution, we have improved our immune system in a very high degree. Actually, one of the main characteristics we have is a very strong and a very active immune system. So normally we speak about this variety of polymorphism as if it were a bad thing. I don't think it is a bad thing. It's just a characteristic. Some of them are very aggressive, but some of them are just protective for specific pathogens. What I mean, for example, here in the slide, you can see that some of the polymorphisms make us very resistant, for example, against coronavirus. Some others, like this one, for example, is quite good to fight against hepatitis or some of other virus. But of course, if you have virus, or if the patient with the susceptibility is attacked by a virus, the immune reaction is going to be bigger or stronger. So now this makes us think that when we have a susceptibility, a polymorphism, which I'm not going to call a susceptibility, I think it's better, or we have to improve our knowledge that these variants or these polymorphisms, this genetic susceptibility or genetic susceptibility actually is a characteristic. Of course, some of them are aggressive, and if we trigger it with different pathogens or a virus or bacteria, the inflammation is going to be very strong. But that also is a reasoning to understand that if we have nice microbes in our intestine, or if we are very healthy in all our barriers, the mouth barrier, the lung barrier, the intestine barrier, we are protected, so we don't express these polymorphisms in a pro-inflammatory, with pro-inflammatory activity. So the most important thing is not the genes. The most important thing is the epigenetic, which is the environmental triggers. So let's talk about it. So as you can see, of course, we can have a strong genetic contribution, but of course we can regulate the immune system even though if we have a polymorphism. One example is that in rheumatoid arthritis, we have this, we have many different aspects that could lead to citrullinated proteins. The main antibody that we can find in rheumatoid arthritis is anticyclic antibodies. And that means that it fights against citrulline. So it's gonna see where citrullinated proteins are, and we can develop an immune reaction to it. Common periodontal bacterium, for example, this one, Porphyromonas gingivalis, and a few others, are able to produce this enzyme, which is called protein arginine deaminase. This is a very exceptional thing because our immune system, the T cells, B cells, neutrophils, and natural killer cells, and lymphocytes, and eosinophils. All our immune cells are able to produce this enzyme when they are in a pro-inflammatory state. So when we have a pro-inflammatory state, we can naturally produce this procedure or this process, which is tainting an arginine for citrulline. So it's a different protein. And when we are converting proteins, the immune system is aware, and the immune system looks at the antigen, specifically the epitope, which is the partial part of the big protein that our immune system will recognize. And that's what we call a cross reaction, or a mimicry, or a molecular reaction to go against our tissue. So what I'm saying in an easy conclusion is that our immune system, if it's in a pro-inflammatory state for too much, for a long time, or in a chronic state, with a lot of trigger in our immune system, or if we have a periodontal disease like gingivitis, then we are going to transform proteins to citrullinated proteins, and the immune system will react against it. Actually, gingivitis is the same as rheumatoid arthritis. If you compare it, we have bone loss in the mouth, and also inflammation, a lot of aggressive infections, and vascular and inflammatory changes, which are very similar than the ones that we have with the same antibody and the citrullinated proteins in both of them. And it's even worse if we have the mouth, the barrier in our mouth. If it's open, then all the bacteria can go and they can disturb the rest of the tissues. But it's going to happen also with some virus, for example. Viruses like Epstein-Barr virus or other viruses are able to induce the PAD, let's just shorten it, the protein arginine deaminase, many of them. So viruses are able to induce PAD in our immune system. So the process is going to be the same, not through the bacteria, because the main bacteria is almost, just a very small amount of microbes has this enzyme. Actually, it's a special characteristic of Porphyromonas gingivalis, this one, but some other things, for example, the damage of the DNA produced by smoking, the damage of the DNA produced with white inflammatory fat or obesity, and all the changes, the metabolic changes in obesity, induces a switch from anti-inflammatory immune system to a pro-inflammatory immune system. That's why in every disease, the amount of fat, the lack of exercise, the increase of microbes, smoking modifies the citrullination. So then our immune system is going to release cytokines, and as much stress, more permeability. So it's going to chronify and increase the process. Then when the immune system is overloaded of different factors and, of course, susceptibility, we are going to start producing antibodies to anti-citrullinated protein antibodies, which are the main antibody in rheumatoid arthritis. And all of them are, when we add some more pathogens or the same ones that produces the inflammatory process, we are going to do a cross reaction. That means that we are going to start to react against our own joint. That's when we call it rheumatoid arthritis. Of course, we can have a seropositive, which is not yet with different antibodies, or many other risk factors, for example, the polymorphisms. So we can have a negative polymorphism and we can have negative antibodies, but we can see that the procedure is growing or increasing, and that's when we call it a negative or seronegative rheumatoid arthritis. And then if you continue in the time, is the time and the days and the years that the person is in a chronic inflammatory process is a higher risk to develop the antibodies. So then we call it a positive, seropositive rheumatoid arthritis. So a molecular mimicry or a cross-reaction is then when the immune system is recognizing our own tissue, and then the destruction of it is higher. Then, of course, we can contribute with different self-proteins. We can attack self-proteins we have been damaged, so they are not exactly the same, cytoskeletal proteins, or even though neuroproteins. So that's why then in the rheumatoid arthritis later on, we can have symptoms everywhere because the immune system is in a low-grade inflammation or a high-grade inflammation in some cases, making a lot more disturbances and symptoms, not just in the joints, but generally. So what we call the movie is that we can have a susceptibility, then environmental factors. Of course, if we open the pulmonar, we can start doing citrullinated proteins because of smoking in the lungs. Then they start making reaction of immune system. This is a very, very studied host modification. You know, the person will modify proteins and they will increase the susceptibility to then having rheumatoid arthritis. As I said, intestinal and mouth permeability in the barriers, not just because of Porphyromonas gingivalis, just also many other different pathogens. So we also have a lot of documentation of when we have invasive microbial disturbance, we also have a problem because we decreases the amount of good bacteria. It's good to remember that we have 1.5 bacteria per cell in our body, and that's just the bacteria. If we also count hundreds of thousands of virus and also parasites or protozoals, some of them are parasites, and also fungus, and also many other microbes that lives with us, and they do an amazing work for us. So good flora produces anti-inflammatory fats. For example, the free-chain fatty acids are, in the intestine, it could be butyric acid, for example, and is a very important substance for the brain and also for the energy and the health of our intestine. And the bacteria produces vitamins for us, enzymes, so we can do digestive procedure, and also protects the immune system. They produce the mucus layer, so we can be completely protected against outside challenges, and many other functions these good bacteria have. So of course, it's easy to understand that if we are a lot more microbes, we have a lot more microbes than cells, then if we change the composition of the microbes in our body, it's easy to understand that the immune system is going to be dysregulated, because we are not doing the function of all this amazing organ, which is our microbes. And but not just the intestinal flora, no, the mouth flora, the lung flora, the skin flora, all of it is all the time moving. So the DNA migrates for all our body, changing our immune reactions. So it's quite important to understand that when we have less diversity, is a lot of functions in our body that we are not performed, they are not good, or we cannot do them. Also, they have a huge communication with our immune system. So it's easy to see that if we have some infections, and also when big or pathogenic bacteria, we have it, the place is already taken. So we cannot have, first we have to clear and to improve and to cure the bad ones, so we can then, with a competitive exclusion, we can introduce the good ones and restore the function. So then when we have a lot of hits in a susceptible person, we are in a second step. We are going to have an immune response, let's call it a second hit, where we start doing like a recognition of all these proteins that have been inflamed and tainted, and we change the order of the amino acids in it. So we recognize them as if it were, because it is actually a bad thing that should not be there. And we start the maturation of the humoral response, and the epitopes spread. Then we have changes in all the proteins or proteins with sugars, the glycosylation process, and we start developing different kind of or an antibody and increasing the PAD that I just mentioned pathway. Then, of course, that means that we need for these epitopes to produce an expansion of the inflammation. And as you can see here, we are going to have reactive, or I don't know, protein C, or C reactive protein. I'm sorry, I'm speaking in or translated from the Spanish. So, that's quite important, because is an inspecific acute phase protein, which is going to try to recover our body against microbes, especially bacteria, but it's also a factor of inflammation. So in every destroyed tissue, we'll have these kind of components. And then we start with chemoxins and interleukins and many other factors, especially from the Th1, which is a cellular against our cells, is a cellular reaction, or Th17, which is a very anti-infective system in our immune system. So in rheumatoid arthritis, the main activation is the immune system that recognizes our own tissue, and also the antimicrobial part of our immune system. So of course, the inflammation and the recognition of self-recognition or the self-tolerance avoidance, because we should see our own tissue and say, this is my tissue, so the tissue is safe. But the problem is that when you develop change in your protein and you attack your own protein, so the self-awareness is lost, and you are going to attack and to increase and increase and increase the immune reaction. So then what we have is a synovial inflammation with a lot of deposition of different proteins and antibodies and citrulinated proteins, anticyclic cyclic, anticyclic citrulinated antibodies, and then immune cells from the T system, the T immune system. So T cell is going to be inside, which is not common. We should have an immunotolerance in our joints. Actually, the joint is quite wide. We should not have neoangiogenesis, which is the increase of the vascular endothelial growth factor. But when we need the immune system to make a reaction in the joint, many other processes which are not physiological start happening, because of course we have a lot of reactive especies reactivas de oxígeno, reactive oxygen reactive species, and nitrogen. So that means that the oxygen is going down, or the breathing or free radicals are increased. So our reaction is to go with vascular endothelial growing factor. We start doing a lot of arteries in order to supply blood and to access with the immune system. But in that moment, the whole physiology of the joint is tainted, because now we are working with blood, with iron, with oxidative stress, and with an oxidative reaction. So now we have a lot of procedures that we should treat, all of them. And of course, then we have to come with a lot more parts, macrophages, neutrophils, and many other types of cells that increase and increase the action. Then, of course, we have an immune cell in the bone. Remember, in the bone, we have two main cell activity. One is the osteoblast, which is his main function is to restore the bone, and the osteoclast, which is actually an immune cell, or is susceptible to be activated by the immune system. So when the immune system, all these factors and join target, of course, the immune system is trying to help us, is trying to recognize things that are not okay because we have pathogens, we have bacteria, we have citrulinated proteins, which is not normal in this degree. So the immune system actually is trying to help us. But the immune activity, if it goes chronic, is too much, so we activate the osteoclast. And at that moment, the osteoclastogenesis will increase the loss of bone, because the osteoclast main activity would destroy, and if it's too long, we have bone loss and many other things. So at this moment, is many others, but in conclusion, what I'm saying is that many toxic and like, for example, smoking, but many others, and things that are happening in the modern life and pathogens potentially contribute to rheumatoid arthritis. At least Proteus mirabilis, Porphyromonas gingivalis, Epstein-Barr virus, and many others have been studied to increase the activity of the immune system, and in susceptible hosts or susceptible patients, they will develop a very huge immune response. But it's nice to repeat as many times as necessary that if we treat the microbes, we will have a good opportunity to reduce the activity, not just the microbes, but many other things. We will have the opportunity to reduce the activity of the immune system. Wait a second, I have a problem here. Okay, it's not a problem. So, then, what we have then is that it's a very special and specific for this bacteria in the mouth, which is mainly when we have a mouth disturbance. Porphyromonas gingivalis produces citrulline peptides, which triggers the immune response, is the targeting of peptidyl-arginine deaminases, and it interrupts the antigens productions and the treatments for rheumatoid arthritis. This is an enzyme that converts arginine to citrulline and is a connection between the porphyromona or gingivitis and problems, disturbance, caries, and plaque, and many diseases in the mouth highly related with joint problems. Of course, it's important to understand that all the good flora in our mouth are improved with, many different things, of course, and we can preserve is more than 700 taxonomic classifications of microbes in our mouth. They are very important to produce digestive enzymes, to produce immune components, and the good ones are quite important to preserve the health of our mouth. And that highly depends on what we eat. So it's very well known in society and in the knowledge that if you eat a lot of sweets, or if we eat a lot of processed food, all the things in our mouth start developing a pathogenic flora. Whenever you eat unhealthy diet, carbohydrates or psychological stress that opens mucous layers and barriers. I cannot touch this, I'm sorry. And of course, a lot of different infections. Of course, when we use a lot of drugs or medication or toxic issues or alcohol or many other substances will induces an inflammatory process. Here you can see healthy activities in the left part of the slide that could improve our systemic health. And it's very, very important what we do with our mouth. And then in the right part of the slide, you can see all the things that are a risk factor, not just for the joint, but also for systemic problems. And that induces the production of pathogenic bacteria. Well, in our body, from any barrier, the mouth, the skin, our lung, the intestine, any of them and more others, the vagina one, many of them, we have different manners, different ways where the flora can go to our blood. So, for example, they can open the barrier because we have a problem, or because you have a wound, or you damage your gum. So if you damage your gum in your mouth, many of these 700 different pathogenic or good ones, whatever, they can come into the blood and systemic circulation and they move very efficiently from our tissues, and they can go in a viable way, in a dormant way, or not viable. So not all of them start going to different tissues in an active form. No, not necessary. You can be years and years accumulating pathogenic viruses, and then one day something else, like a drop in a glass of water, there is like one issue. When you have summarized, when you have add a lot of drops, then one day the inflammation comes very aggressive. Why is that? So as you can see, this kind of bacteries will start going and doing a process, and one activation and the other one. Then you start producing antibodies, you start having a small pain, but then it starts involving a lot of different cardiovascular diseases. The metabolism starts to fail. The metabolism also increases, and the activity of the immune system, and you are susceptible for many other systemic problems for the same unbalanced microbial distribution. So that makes us susceptible and more weak to fight against lot of other disease. So mouth is a priority here, is a priority to be careful in these patients, but also for adverse pregnancy outcome, or to increase the susceptibility of diabetes, or to neurodegenerative with all these microbes related to the mouth health. For example, for many other, not just Alzheimer's disease, but it's a very well documented how the mouth can increase neurodegenerative diseases and many others. It's important then to explain that we can then increase the amount of pathogenic bacteria with all the bacterial mechanisms to induce immune dysregulation and to induce metabolic changes. So now everything is more aggressive and the inflammation is more aggressive, but also it's important to understand that when we have a damage in a tissue, we release iron. The microbes are very needed. They need iron and sugar and some other things, but especially iron, to develop and to proliferate. And actually, they are a lot more virulent and a lot more aggressive in the presence of iron. So what I said before is that we can have it in a dormant state. They are like very in low activity, they are methylated or they are not doing anything, but we are increasing and year by year by the permeability, the amount of microbes is higher. And then one day we have a hit or a trauma, or something that destroys the tissue, the iron, because of the small hemorrhoids, or just because of the permeability increase, and then this iron is taken by the microbes and they start being aggressive all at the same time. So that's why sometimes you can see that a person has an accident or a trauma or an emotional trauma that opens the barrier, or they don't take care of the hygiene of the mouth or the safety of the barrier, or, you know, to have a very good oral health, and then suddenly they start with a very aggressive inflammation. But that's because they accumulate a lot of microbial and immune activity, and then one day is in a acute state. So here we have many different things that have sum up until we start having the problem. So hingipains are also iron binding that breaks down ferritin and transferrin and other iron-containing proteins molecule, and then the oral mucosa will open and the viable bacteria will become active. The ones that were in an unviable or in a dormant state. So they become aggressive whenever they have iron ability. As you can see, each bacteria has its own cytophore, like boomerang proteins that binds iron. So different bacteria, here you have some of them, and related type of disease that we know that these pathogens are related to this disease, and then they have a special structure to use iron. This is very important. I'm going to speak I'm going to speak slower now because this is a very clinical important issue. Why? Because in many cases, immune patients are with anemia, and actually they have recurrent anemia, which means that the iron decreases, and also some of the iron storage proteins. In many cases, because they feel tired, or all the symptoms of a lack of iron, in clinics or clinicians advise iron to these kind of patients. But if I just said that the iron increases the inflammatory process and increases the aggressivity, the virulence, and the replicability of the microbes, it's quite important first to treat the permeability, treat the amount of microbes we have, and to heal all these barriers, and then we can restore the iron amounts. But actually, this is a normal immune reaction to develop to eliminate iron in order to preserve the host health. So it's good for the host. It's important that we are careful in the iron prescription in all the immune diseases. So this is quite important. Chronic pathology is configured, is produced through the entry of pathogens. And one of the good things we can do is to do a chelation of iron. There are a lot of possibilities. For example, one resource that you have in India is curcumin. Great curcumin you have. So curcumin is a chelator. Is something that is very helpful. I call it the golden spice. It's also like gold, the color of it. And it's very important to understand that it's good to do, to eliminate or to bind iron, to eliminate it. So it's good. So our immune system can, we all know, or I'm going to mention that curcumin is very, very evidence-based for to be anti-inflammatory, but it's also working in this mechanism. It's also a very good antiviral that helps a lot because it's a lot of virus in the activity of the Th1 system, which is this system, is the recognition system. So we can clean and we can, it's a good natural treatment for virus and also as a chelator. But for sure, lactoferrin, which is a transferrin of our immune system, actually we produce it doing exercise. If you do exercise, you induce in your neutrophils the production of lactoferrins. Is the main defensine of all the mucus layer in all your barriers. Is a very important to eliminate and to promote good flora and to diminish or decrease the bad flora, but also is a very nice chelator, and it's bacteriostatic. Also, you can add lactoferrin to a bacteria in order to eliminate it and recognize it. You can also bind or take few lectins and eliminate them. There's many good things in it. You really expect you read about this substance because it has a lot of immune different kind of functions. And it's also very good as an antibacterial, antiviral, anti-microbes, all of them, for the good flora, and it's also organizing the iron system. The good thing is that because it's a transferrin with 4 molecules of iron, you can give it for anemia. It's a very good to erythropoiesis, is a very good to decrease immune activity, is an anti-inflammatory substance, but it's also promoting the protein availability of iron. That means that your immune system and your cells can use it because it goes in a protein, it's a storage protein, but the bacteria or the virus or all of them will not take it. So it's a good solution for also the tiredness and the immune activity. So we are talking that is then the intestinal or altered permeability that induces the amount of bacteria that goes to normal or systemic circulatory system. Then we have a lot of substances from endotoxemia, means that we have a lot of negative gram-negative bacteria. They have one characteristic, they pour kind of a protein which is called lipopolysaccharide, and the amount of it will extremely activate the immune system in many different parts, recognized by toll receptor system, and then is going to increase the inflammation. No, just because the bacteria are going into circulation, even though if we attack them, these parts, these lipopolysaccharides will induce that we continue doing inflammatory processes. So then we have a lot of significant substances of immune system that trigger abnormal immune responses. As I said before, it's important to understand that we have a lot of bad bacteria increased in this pathology in rheumatoid arthritis. Here you can see some of them that are increased, but also we have decreased good flora and permeability. So now we know that the composition of a leaky gut is also performed or induced by the dysbiosis or dysbiosis, which means that we have bad pathogens in our intestine, but also in many other tissues. Here you see the autoimmune diseases related to low-grade inflammation, leaky gut, and dysbiosis or dysbiosis. Okay, and we already know that in multiple sclerosis, which is very similar and actually is the same anti-citrullinated antibodies, is the same antibody of arthritis, rheumatoid arthritis. And it's very important this because when you have one pathology and is rolled, it's like performed by the same immune reaction, in years you are susceptible because these antibodies are going in systemic circulation all the time. So one day they can get sensible to substances that you have in your brain, and you will also have other immune activation in other tissues, for example, multiple sclerosis. So if we have an amount, now we know that we have sonulin, which is a protein that expresses that our intestine is permeable. This is a protein that guarantees that we are very connected from one enterocyte to the other, and is protective. And if this protein is in a high amount, we know the destruction of the tight junctions and many other systems that preserve the physiology of the intestine is damaged. Okay, so this that you have here is like the way that we can see, for example, the amount of lipopolysaccharides or the amount of proteins. That means that we have like destruction in our intestine. So now we know that for all the immune disease, the basic thing in it is that we have intestinal permeability. So of course, now we know some of the bad bacteria, we have them clear. It's not completely studied this, but some of them are very well described. So here then you can read more calm that is not just immune diseases. Of course, depending of the characteristic of each person, we can develop in a susceptible Th1 immune system or with immune polymorphisms, is a lot more probable that we express or that we end up with an immune disease like rheumatoid arthritis or a different one, but also is highly related with allergies, rhinitis, atopic dermatitis, or sinusitis. And that means that we have a different condition or a different amount of risk factors that induces a change in the Th2 system. So leaky gut and bad microbes will induce a lot of different pathologies in the immune system. Of course, we can measure it. I think I'm running out of time. We can measure it. You have different kind of exams. Here you can read some of them, or you can search for it. No problem. This is recorded, so you can read it later on. We have different lot of different permeability tests, so we can do a diagnostic. But don't worry, because if you have a bleeding gum, you have symptoms, because it's important to say that we have tests, we can test it, antitrypsin, protein, and sonulin, and many other things we can test it with clinical medical exam. But it's also important to remember that semiology, or the study of the signs and the symptoms, is the highest level in science to understand what's troubling or what's a problem of the patient. So if we have a lot of pain, inflammation, and immune disease, and also the behavior of going to the bathroom and all the shape of feces, and we have many, many markers to understand that the person has permeability. But anyway, we also can test it. Then, of course, when we treat an intestine, then we need, or we can do a lot of different tools to restore the barrier function and to induce good flora, that the good flora is restored too. So now we know that, of course, probiotics, prebiotics are very, very nice diet and glutamine and vitamin D, and specifically good healthy food and healthy habits would definitely improve the mucus layer, and also the well, in some cases with a lot of interest and knowledge about nutrition, we can restore a lot of things. And also we can decrease the pathogenic bacteria, or at least improve the immune system, so we can improve the immune system, so the immune system is capable again to fight against these infections. We are very, very, very efficient in organizing our own microbes. Wait a second. So it's important also to understand that nutrition is a lot more important than we suspect. It's not just because of the ability of micronutrients and vitamins and polyphenols and antioxidants and all of this that everyone knows. It's because there are some nutritional factors, like corn, like gliadin or lectins in cereals, or lectins in and agglutinin in gluten, and also in wheat germs and in peanuts, that can produce a reaction to these antigens. So these are strange proteins that when you have permeability, these nutritional factors includes a lot more proteins, lectin proteins, for example, and your immune system will recognize them, increasing the pathology or the immune activity. So some nutrients, of course, sugar, peanut, corn, a lot of different legumes, a lot of different special refined or not processed food, induces an immune reaction to our tissue, or a cross reaction, or a molecular mimicry. So there are environmental and nutritional factors that also contribute with autoimmunity in various ways. That means that we have a molecular mimicry or a cross reactivity and the formation of neoantigens through the binding of toxic substances and proteins in our tissues. Here you have sequences of these proteins, for example, gliadin in gluten, in wheat cereals, in bread, in cookies, in a lot of different grains that contains gluten. And we know that this protein in gluten, this protein in cereals, is very similar that proteins in our cells, in our tissues. So in an activated immune system, or when you process this food, when you technologicize this food, we can do a cross reaction because of a nutrient. And this is quite important because we know it's an elevated immune reaction to proteins such as milk, wheat, contaminated eggs, and especially pork. What's wrong with egg and fish? Well, that's not very normal, but because we have plastic or toxins or pesticides in few nutrients, we will start doing allergies to food that did a very evolutionary coexistence with us, because of course anthropologically eggs and fish is part of our nutrition, and the problem is that we are transforming with technological procedures and ultra processed food. We are changing the proteins in food, and there are some toxics, lectins in food, or anti-nutrients in food that could cause the IgM antibodies against anti-immunoglobulin G, which is called rheumatoid factor. Why I say this? Because rheumatoid factor is something that we measure very, very specific of rheumatoid arthritis. And the contribution of nutrition to the increase of the inflammatory response through rheumatoid activity, we can treat it changing nutrition, or we can decrease it, or we can modulate it. This is quite important because the tolerance starts in the belly. We can start pushing antibodies and also immunoglobulins that has react to nutrition or to microbes through the baby. And actually the placenta is quite protective, but there are antibodies that can go through the placenta, and then the babies are already susceptible for disease. So the loss of tolerance can affect oral tolerance or intestine tolerance, will affect the health and the immune system of the pregnant mother, but also to the child. So it's important because the dietary peptides, epitopes, which is this small sequence in a antigen, shares homology with human tissues. So since the baby is born, the baby can be susceptible because of nutrition for immune reactions. And this is something you see in the clinic, because now we have a huge increase in dermatitis, in skin problems, in lung problems. Just one generation ago, just 20 years ago, it was a lot less prevalence or the incidence of allergic disease or immune disease or skin disease in kids, even allergies, was a lot lower. Now in some countries, we have one every 4 or 5 babies that are suffering because of contamination and nutrition and some other factors for inflammatory and allergenic diseases. So it's quite important. I'm sorry, I think I'm quite late, but it's very important that we have evidence about a lot of different cereals and grains that can cause cross-reaction with gluten, and especially processed food is highly effective to activate permeability and to induce an inflammatory immune system. The second thing is that we have in milk, in cow's milk, a lot of different proteins responsible for a huge immune reaction. And it's mainly because of all the transformation we do, that of course, if we consume and not industrialized product, which is a road, which is completely natural for a health wild one, the reaction will be completely different. But as we are taking nutrition right now with the industrialized food or ultra processed food, is very important factor to activate the immune system. We have a lot of different evidence that especially red meat, that I know in India you don't need a lot of it, but red meat and grains, wheat germs, these are the maximum reactivities we have for rheumatoid arthritis. So we know the peak is a purish and almost every mammal, because of sugar that the mammals has, which is called NEO5-DC. Also, pig is a highly reactive nutrient, the wheat germ, and also the cow. And it's quite important that we have cross reactions also with corn and other grains. Of course, we have many other environmental factors. We have fertilizers, we have pollutions, we have pesticides, herbicides, and of course that increases the susceptibility of the immune system. And you will say, yeah, but we have a huge country with a lot of population and a lot of contaminants. Yeah, of course, but of course we can handle it. Even though you are in a place with a lot of, I don't know, heavy metals or pollution, it's important to understand that when we sweat, when you do a lot of exercise, when you go for a sauna bath, when you do whole body heat and increase the sweating, you increase the ability to eliminate some metals that you cannot eliminate through any other pathway or mechanism. So it's great to understand that nutrition has an amazing way to be part of a treatment, but also movement, but also lifestyle changes. So I add here a few interventions. We are strong in addition to regular treatments that we do, and it's very important to handle nutritional, psychosocial stress, understanding the movement avoidance is chronifying and focusing in the problem is very unhealthy. If we focus the patient to many different solutions, for example, to increase the omega-3 in algae and in fish food, the omega-3 is highly anti-inflammatory, eliminate all omega-6 in vegetables, oils, that would definitely balance the immune system to anti-inflammatory condition. Also, here you have it, the omega-3 is highly anti-inflammatory. If we decrease the amount of cereals, processed food, and technology-sized food, and food that is preserved for a lot of time in a bag, is high in the content of omega-6, so we can reduce the consumption of omega-3, and also, of course, we need to reduce the mammal, and that's quite important to reduce the antibodies against the in rheumatoid arthritis, but all the mammals meats will be a very great strategy. For sure, there are a lot of evidence polymorphisms about how much vitamin D we have and how much we can metabolize. So these kind of patients really need to go daily for sunlight because of vitamin D, and the sun exposure, but daily, not intermittently, is every day to be very tanned and outside and in the as much as we can in nature, is a highly efficient technique to produce vitamin D. Vitamin D targets more than 167 genes to the anti-inflammatory immune system in a direct or an indirect way. The vitamin D is a neurohormone, because actually vitamin D is a pro-hormone. So it's very important to change the immune system. And for sure, the muscle is incredible. How much different mechanisms to improve our health we can do with the muscle. So of course, we know at least that exercise trigger a lot of different neurotransmitters related with the good immune system activity. It also neuromodulates through endorphins and many other analgesic or anti-pain substances. The myokines are released by the muscle, and they regulate the immune system switching the immune system to an anti-inflammatory, but it also induced the production of anti-inflammatory cytokines and defensins. I said an example before. I said these myokines can induce the production in our barriers of lactoferrin, for example. One of the main issues is that when we do exercise, and if the rheumatoid arthritis patient is suffering a lot of pain in their knees, it doesn't matter. We can do exercise in our upper body, or we can wait until they decrease the huge inflammation process, and then we can wait that and then start little by little improving the muscular ability and muscular flexibility with different kind of exercise, depending on the muscular fibers that we are using. We can actually compete with the immune system. So physical activity maintains the insulin sensibility, regulates metabolism, and please remember that glucose and LDL cholesterol, for example, the lipid profile, are two main players to induce inflammation in the immune system. So what we call bad cholesterol, if it's too high, is a molecule that will increase the activity of the immune system. It's immunogenic. So it's quite important that we understand that we have to regulate in these patients that a lot insulin growth factors, glucose, and we have to restore the lipid profile. It's quite important. They are thin and without visceral fat, so we can improve the intestine, but also the metabolism that for sure organize the immune system. So is the metabolism handling the immune system, and also the brain handling the immune system, and for sure the barrier if we restore with nutrition, exercise, and some fasting, if it's possible, we can then decrease the activity of the immune system. For sure, the psychosocial stress is highly evidence-based to accelerate symptoms and, of course, induce pain. In these patients, it's quite important to understand what's happening in their life, try to do amazing, beautiful things if possible, and also integrate them because they have a problem with to be capable to do what they wish. So little by little, avoid catastrophizing and try to give them a focus in solution to improve the quality of life. Just a small thing is important. A small summarize is amazing the results we can have using a high amount of polyphenols, which has, for example, curcumin, resveratrol, green tea, and a lot of different mushrooms, herbs, and spices. You have the great amount of them, and reach a huge several things that you can. That you have a different amount of spices as a variety. So what I mean is that if you use a lot more variety and diversity, you have a diversity of polyphenols and you will improve your treatments. Then omega-3, for example, C derivative nutrients, increase intake to support the immune system, reduce mammal consumption, and cut out dairy derivatives. This is quite very important. Avoid ultra-processed food, do exercise, and eliminate sugar consumption. This is very important. So lifestyle adjustments, dietary choice, smoking cessation, optimization of lipids, increasing vitamins profiles offer a pathway to improve prognosis, but also it could really, I worked with immunology since 23 years ago, and sometimes it's even better than we expect. It's a very aggressive activity for the joints, and it's important to work in as much areas as we can to rehabilitate and to recover the patient. So rheumatoid arthritis is a complex interplay for genetic predisposition, environmental triggers, hormonal dynamics. So working with nutrition, cleaning your mouth, eating properly, doing coconut oil hygiene in your mouth, and especially doing exercise, decreasing the amount of sugars will, of course, reduce the aggressiveness of the microbes, and then you have specific treatments for microbes, which you can also use. Well, thank you very much for you that continue here. And I hope if you have any question, I'm available for you.
Thank you so much. We are open for questions if you have any. I think the lecture was pretty self-explanatory and you did a great job, especially the topic was new and it was a difficult topic to deal with, and trying to comprise so much of information in such a short duration of time definitely becomes very difficult. But it was a great lecture. Thank you so much.
Thank you. Thank you for the invitation, and I hope you can, take some advices to do with the patients in the clinic.
Yes, I hope it was a very helpful lecture. So I would like to propose a vote of thanks. So gratitude is not only the greatest of the virtues, but the parent of all other. And therefore, I, Dr. Zubia Wakar, in front of you, would like to propose a vote of thanks on behalf of the organizing team. First of all, my heartfelt gratitude to our speaker of the day, Professor Maloha Casanova Mendes, for not only sparing her time for us, but also to deliver this invigorating lecture, which has ignited our minds. Thank you, ma'am. It's indeed a great honor for us to have you welcome as a prominent speaker who is known for her great contributions to research. We definitely feel enlightened and believe that we got to learn so many new things from this lecture. Words, I think, definitely would fall short if I want to describe how much knowledge we have derived. Thank you so much for being a source of knowledge for us today.
Thank you. I hope to see you again, and I hope to be in India soon. So
yes, definitely looking forward to seeing you in person in India.
Thank you for all the team, because we are a team in PNI Europe. So we really appreciate your invitation, and we hope to come as a team again.
Yes, sure. And I would also like to propose my vote of thanks to Professor Sanjeev Kumar Jha, the IAP president, for his constant commitment and support towards the profession. Thank you, sir. Our Vice President, Dr. A. Suresh Babu, has always been a pillar of strength. Thank you, sir, for your constant support and encouragement. Dr. K. M. Annamalai, Secretary IAP, has been a source of guidance. Thank you, sir, for your time and your vision. My heart goes out truly to Dr. Ruchi Vachne, Treasurer IAP, who's a constant guiding light to all of us. Thank you, madam, for always being our guiding light. Next, I would also like to thank all our CEC for being our constant support systems throughout the time. Whenever we are looking to organize anything at a zonal level, at a state level, they are the ones who we are looking up to. Thank you from the bottom of our hearts. When we have reached the end of it all, it gives me immense pleasure to see the proud faces of the IAP digital team members who have worked tirelessly throughout, as well as before the lecture. Thank you so much for your tireless efforts, without which it would not have been possible for us to have such a great webinar. Last but not the least, I would also like to thank all the participants who took out time from their schedule to attend the webinar and made it a great success. I hope all of you have learned something new, as I have. Thank you so much for listening. And we thank you all for being here with us this evening. It has been a pleasure. Thank you once again. Thank you so much, madam. We look forward to seeing you in India soon.
Yeah, me too. Looking forward to travel there.
Absolutely. We're looking forward to welcome you to India.
Bye-bye.
Bye-bye.
Bye, ma'am.